In the 1990s, a new technology came up called marker-assisted selection, where you do away with the phenotypic evaluation, that is looking at the plants in the field and deciding what the characteristic is, or by inoculating with the pathogen and finding out whether it is resistant or susceptible. So this technology was, you would use DNA marker technology to identify within a large population of plants which plants have the desired characteristics. So we developed a elaborate breeding program where we use DNA markers to identify those plants that have the bacterial blight resistance characteristics and those that have likely to have the yield and quality of Samba Masuri. This we did over several generations of crossing and ultimately came up with this product called improved Samba Masuri that has bacterial blight resistance and has the yield and quality characteristics of Samba Masuri.
ICAR funded this research project under its ambitious National Agricultural Technology Project, whereas CSIR Lab got the financial support from its CSIR 800 program, which is a flagship project aiming the socio-economic enhancement of 800 million Indians who live in rural areas or constitute the urban poor. Scientists adopted strategies to improve Samba Masuri rice to make it resistant to bacterial leaf blight disease in the improved version by gene marker technology or marker-assisted selection. Marker-assisted selection is one of the most important and latest technique used today in development of new varieties. So DNA, RNA that carry specific hereditary characteristics are selected through marker-assisted selection, and such markers from different plants are sampled or incorporated into one. This is a long process which requires series of steps carried out in laboratory where researchers deeply study. But the challenge was to keep the desirable traits of Samba Masuri intact. Dedicated team of scientists of both institutions initiated work in the sophisticated laboratories in 1999. Latest tools and cutting-edge technologies were used to get best results. The new variety thus developed was aptly named as Improved Samba Masuri ISM, and laboratory tests were conducted to observe and evaluate its characteristics and yield.
There are trained personnel or experienced personnel at the Indian Institute of Rice Research who can tell whether a variety is Samba Masuri or not Samba Masuri by just tasting it. So we cooked Samba Masuri and improved Samba Masuri, gave it to them, and they could not tell which is Samba Masuri and which is improved Samba Masuri. On that day, we thought that we had achieved success in our project.
After getting satisfactory results, field trials started in 2005-2006 in multi-location settings across the climatic zones. Field trials were conducted for 2 years in 10 selected locations across the country under the supervision of All India Coordinated Rice Improvement Project.
Okay, we have developed improved Samba Masuri. It has now gone through the national field trials. Indian Council of Agricultural Research had tested it rigorously, and they had indicated now that improved Samba Masuri was bacterial blight resistance and had the yield and quality of Samba Masuri.
ISM variety proved its worth in field trials by showing resistance to bacterial leaf blight and retaining all the good characteristics of its predecessor variety, Samba Masuri. A new era was just to begin. On the recommendations of Scientific Committee, Ministry of Agriculture, Government of India notified ISM in 2008 for commercial cultivation in several states, mainly in undivided Andhra Pradesh and Karnataka. Subsequently, the variety was also registered with the Protection of Plant Varieties and Farmers' Right Authority (PPV) and FRA, Government of India, to protect the right of plant breeders. Come and be a part of this thrilling and exciting journey as how science has changed millions of lives.